Chromosomal inversions. Chromosomal inversion is two breaks within a chromosome and the fragment of the chromosome inversed. If the segment of the chromosome is in this direction, it will be reversed during chromosomal inversion. Chromosomal inversions can be of two types. One is called as pericentric and the second one is called as paracentric. In case of pericentric inversion, centromere is involved, while in case of paracentric, centromere is not involved. In this diagram, we can see that difference between pericentric inversion and paracentric inversion. In case of pericentric inversion, the inverted region, it contains the centromere. This is the centromere of the chromosome. If centromere is included within the inverted region, then this type is called as pericentric inversion. While if there is a inversion of the segment and the centromere is not included, like in this case, then this type of the inversion is called as paracentric inversion. In most of the cases, there is no loss of the genetic material as a result of inversions. In those cases, there is no consequences to the phenotype. But if the inversion is at a point where there are the promoter sequences of a specific gene, if the break is from that particular point where there are the promoters present of the gene, then it may have effect on the phenotype of the individual. In case of chromosomal inversion, most of the cases, just the position of the chromosomes is changed or the position of the particular fragments of the chromosome is changed. It goes from one orientation into another orientation. Approximately 2% of the inversions can be seen in case of humans by a technique which is called as karyotyping. Heterozygote individuals in case of inversions are those individuals that contain two chromosomes. In one of the chromosome, they have inverted segment of the DNA, while the second chromosome that is has normal sequence of the DNA or the normal positions of the genes. With this diagram, we can understand that what are the inversion heterozygotes. In the first diagram or in the upper diagram, we can see that this is a normal chromosome, but in this case, the fragment of the DNA has been inversed. So here the sequence has become A, E, D, C and B. Originally it should be A, B, C, D and E. So this is inversion in one of the strand of chromosome while the second strand is normal. So if one of the strand has inversion and the second strand do not have inversion, such inversion is called as heterozygote, and the individuals, they are called inversion heterozygotes. In case of where there is centromere involved, in case of the second diagram, here we can see in this case centromere is involved. This is the centromere. In this fragment, centromere has been removed, has been deleted. So this is acentric fragment, while on the other hand, there is a dicentric chromosome that consists of two different centromeres, centromere 1 and centromere 2. So as a result of inversion, centromeres can be lost and centromere that can join within a single chromosome. So these are the examples of the inversions. Inversions can be without the centromere by including that centromere within the fragment that has been inversed. Most common inversions in case of humans have been seen on chromosome 9, where a fragment from arm P is changed with the fragment that is on arm Q. Generally, these inversions in case of humans, they are not harmful, but they increases the risk of miscarriages.